Welcome back to my channel, and today we are going to discuss about the different theories of apical meristem. So first of all, let's start with the apical cell theory, which is the oldest theory, which was proposed by Nagali in 1858, and it was proposed to explain the organization of apical meristem in case of lower plants. In case of lower plants. And according to this theory, the growth of plants' apex is controlled by single apical cell, which is also known as the apical initial, and this single cell is all in shape, that is a pyramidal or vase shaped cell, which is located at the tip of a root or shoot, and it divides in 3 or more planes, giving rise to all the tissues of the plant body. The theory holds true mainly for algae, bryophytes, and some pteridophytes, where a single apical cell governs the entire meristematic activity. However, it is not capable to the higher vascular plants, where several cells participate in the apical growth. So, after all this, in short, you can understand in simple way that the apical cell theory was proposed by Nagali, in which it is said that a single apical cell or the apical initial, which is pyramidal or vase shaped, is located at the tip of stem or root that divides in 3 or more planes, giving rise to all the tissue of the plant body, which is applicable to the cryptogams actually, which whereas it is not applicable to the higher vascular plants like phanerogams, and because there are several cells that participate in case of the phanerogams in the apical growth. So this is a simple figure where you can see a pyramidal apical cell. This is the leaf primordial, and this apical cell is all from which the other give rise to all the tissues of the plant body through its division. That is the derivative of the apical cell. Now let's move to the second theory, that is the Histogen theory, which was proposed by the Hanstein in 1868. According to this theory, it explains that the apical meristem, that is the apical meristem of the higher plants, in case of higher plants, is differentiated into 3 distinct histogens, and the 3 distinct histogens has been given the name, that is Dermatogen, Periblem, and Plerome, and each of the following has its own function. Actually, each histogen give rise to a specific tissue system. That is the dermatogen. Forms the epidermis. Dermatogen forms the epidermis. You can see here dermatogen, E for epidermis. Likewise, the periblem forms the cortex or the ground tissue, and the plerome develops into the stele or vascular tissue. So here, dermatogen forms the epidermis, periblem forms the cortex or the ground tissue, and the plerome develops into stele or the vascular tissue system. And this theory emphasized cellular specialization and functional donation in the apical region. Let's look at the schematic diagram of the shoot apex according to the Histogen theory. In the higher plants, the histogen have been divided into 3 distinct layers, that is dermatogen, periblem, and plerome, where each of them has the specialized tissue system, give rise to specialized tissue system as mentioned above. Now third one is the Tunica-Corpus Theory, which is the most accepted explanation for the organization of the apical subapical meristem, which was proposed by Schmidt in 1924. According to this theory, the apical meristem is divided into two zones. You can see here the apical meristem is divided into apical cell is divided into two zones, that is tunica and corpus. In case of tunica, it may be comprised of layer 1 or sometimes more layer like layer 2, and that divides anticlinically. In case of like ta, that is tunica is divided anticlinically, that is perpendicular to the surface, and that helps for the contributing to the surface growth. So like you can remember like the TS transverse section, in that way tunica for the surface growth and anticlinical division thus. Okay, and the next one is the corpus, where it is lying beneath the tunica. You can see this is a simple figure of the apical meristem. Here the tunica may have a single layer or sometimes double layer. This is the tunica, and below, which is for the surface area, surface growth, or contributing to the surface growth. And formation of the epidermis, as it has the, this is for the formation of epidermis. And in case of, this is the corpus, and in case of the corpus that is lying beneath the tunica, it consists of the mass-like cells that divide in the various planes, adding to the volume CV. You know, it's very important, CV. So for the corpus, it is for adding the volume and the internal tissue of the plants, and this theory successfully explains the pattern of cell division and tissue differentiation in the pseudapex of higher plants. So after all this, let me revise for you all in a very short way pseudapical meristem theories. SAM. First is the apical cell theory, where a single apical cell, or the pyramidal or wave-shaped, that is also known as apical initial, can lead to the development of entire plant body. Next one is the Histogen theory, where the histogen in the higher plants have three distinct layers, that is Dermatogen, second is the Periblem, and third is the Plerome, where the Dermatogen leads to the formation of epidermis, that is D for E. D, Dermatogen, for the formation of epidermis. Periblem, that is for the ground tissue, hypodermis. Cortex, endodermis, and the other is the Plerome, that is for the vascular cylinder in a simple way, that is xylem and phloem. And the most accepted theory that I have told you is the Tunica and corpus theory, where tunica is the outer, which has anticlinal division, that is help in the formation of epidermis, which may be one or two layer. And corpus is the inner to the tunica, that is helping in the formation of cortex and stele, which is the most accepted theory. So that's all for today.